Hello everyone, my name is Louise Camus. I'm currently a PhD student in Montpellier in France, so that's why I couldn't be here with you today. But I'm still going to present you a case study on the fruit pest Drosophila suzukii to discuss with you how we can predict the invasiveness of a species with genomic data. So, as you all probably know, the genome of living beings allows them to adapt to their environment. So this environment can be as well biotic, such as the type of plant host for fruit pest, but also abiotic, such as the temperature or pesticide use, for example. And so something that is great is that nowadays, with recent sequencing techniques, we are able to sequence a lot of individuals and therefore characterize the genomic composition on a population scale for population that live in different environments. So in other terms, we are able to modelize the variation of allelic frequencies according to the environment, which can be characterized by variables such as temperature, for example. And so this genome environment association is very important because it can allow us to identify markers or environmental variables that play a role in local adaptation. And if this association is well done, it can theoretically be used to do some prediction. For example, if you have a population that lives in a given environment and that the environment of this population changes, so we move along the x-axis, we are theoretically able to predict the allelic frequency change that is according to the model needed for the population to adapt to this new environment. So this is this y-axis change, and this theoretical allelic frequency change is actually called the genomic offset. And this is really a measure of the level of maladaptation, because the higher the genomic offset is, the higher the risk of maladaptation to a new environment is. So this genomic offset can be either temporal, meaning that it can be computed for future climatic conditions, or spatial, computed for conditions in other geographic areas. And so this genomic offset index could be particularly interesting for invasive species because it could be used, for example, to predict geographic areas that are more at risk to be invaded, or a type of plant host that are more at risk to be destroyed, for example. But it's not that simple because, first of all, many methods can be used to compute genomic offsets, and these methods have been very little validated in literature. So one main objective of my PhD is to evaluate some of these methods and try to improve them if possible by using simulated data and also real data from population, natural population and laboratory population for two invasive insect species, which are Drosophila suzukii and Harmonia axyridis. And the second objective of my PhD is to apply those methods to invasive species because for now, to our knowledge, it has not yet been done. And so this is what I'm going to talk to you about today by showing you results of analysis that we made on natural population of Drosophila suzukii, which is an invasive fruit pest. And so we did this analysis to try to better understand invasion. So we tried to answer two main questions. The first one was to identify which variables were more related to invasion, meaning which variables are more important in local adaptation. And we also wanted to determine to what extent the genomic offset was a good indicator of invasive potential. So we first computed genomic offset of native population from an environment different from the one they originated from. And then we asked ourselves if the genomic offsets we computed were related to what we knew about Drosophila suzukii invasion. So we expected that first invaded areas would tend to have lower genomic offset than late invaded areas because population would have adapted easier to these first invaded areas. But so what do we know about Drosophila suzukii invasion? So this species is native from Asia, and we know that it has invaded almost simultaneously and independently the American continent by the west coast and the European continent by the south. So we used sequencing data from 43 populations distributed in these three areas which allowed us to identify a bit more than 3 million SNPs. But as our goal was to better understand adaptation to environments and that we have two independent invaded environments, we decided to create two SNPs datasets for each invasion route by selecting SNPs based on a contrast index. So this contrast index is very simple. It simply tells you how much the allelic frequencies of a SNP are contrasted between two groups of population that you choose. So we first created a dataset with the 10% SNP that are the most contrasted between native and invasive European population. And then we did exactly the same, but this time with the 10% SNP that were the most contrasted between native and invasive American populations. Then we associated those two allelic frequency datasets separately with 19 bioclimatic variables with the machine learning approach called gradient forest. So this is an approach that is based on random forest, but I won't go into much details about the method here. The only thing you need to know is that one of the main outputs of these methods are these types of curves, which are called turnover curves, that are outputted for each environmental variable in the model. This curve tells you how much the allelic frequencies of all your SNPs changes according to an environmental gradient. So to give you an example, here you have an environmental variable, which is the precipitation of the warmest quarter. And you can see that between 0 and 20 millimeters, the slope increases very sharply. So this means that between those two environmental conditions, there is a lot of allelic frequencies changes. On the opposites, you can see that between 600 and 800 millimeters, the curve is almost flat, meaning that between those two environmental conditions, the allelic frequencies almost doesn't change. So these curves are outputted for each variable, and they also give you information about how important a variable is to explain allelic frequency change. The higher the curve goes, the higher the variable is important, the most important the variable is to explain allelic frequency change. And so these turnover curves are what is used to compute the genomic offset simply by computing the y-axis difference I was telling you about before for each of these variables, so each of the turnover curves. And then we sum up all of those y-axis difference by weighting them with the importance of the variable, and you can obtain a global genomic offset. So now, what are the results? So we wanted to understand which variables were more important in local adaptation to invaded environments. So these are the results of the importance of the variable for the European model, meaning the model that was based on the European SNPs, so the one that were the most contrasted between native and invasive European population. So here you can see that the most important variables to explain allelic frequency change are the precipitation of the wettest months, the precipitation of the wettest quarter, and temperature seasonality. Now, what do we have for the American model based on the other datasets of NIP? So you can see that the important variables are not the same. Clearly, the most important variable is isothermality. And in a lesser extent, you also have two variables related to temperature that are relatively important. So with this, we have our two models and the importance of each variable, so we can compute genomic offset. So here, what you see is the genomic offset, which is computed with European model between environmental condition in Beijing, which is a native population, and environmental condition in Europe. So you can see that overall, the genomic offset is very low in Europe because the color is mostly pink. And the lowest genomic offset area is actually Eastern Europe, which is not yet fully invaded to our knowledge. But we can also notice that the first invaded areas have relatively low genomic offset. On the opposite, very high genomic offset areas are, for example, Norway, which is not yet invaded to our knowledge, or Ireland, which is one of the latest invaded countries in Europe. So now what do we have for the American model? So this is exactly the same thing, meaning the genomic offset between Beijing and environmental conditions in America. So the first invaded area has mild genomic offsets, but clearly the lowest genomic offset is actually in the north of North America, which is not yet fully invaded to our knowledge. And on the opposite, a very high genomic offset area is Central America, which is not yet invaded to our knowledge. So what can we say about those results? First of all, we've seen that the important variable to explain local adaptations were different depending on the continent. So this shows that there are probably a different adaptation basis for invasion, which underlines the interest of genomics. Also, we've seen that the genomic offset was consistent with the history of invasion of Drosophila suzukii, because none of the first invaded areas have high genomic offsets. Every high genomic offset areas are either late invaded or not yet invaded. But however, we've also seen that some areas with the lowest genomic offset are not always the first invaded and are not always invaded. So this probably simply shows that to invade, it's not the only thing necessary is not only to adapt, but it's also other factors such as, for example, accessing the area, for example. So then what do we want to do next? As we've seen that some variables were more important than others, but those variables are the most important one amongst the one we've put in our model in the first place. And also we've seen that other factors can favor invasion. So it could be very interesting to take in account other variables in the model, such as pesticide use, for example, or also the wind that can facilitate probably the movement of individuals. We've also seen that the genomic offset is consistent, which is very encouraging, but these remain very empirical results. So that's why we are currently building simulation scenarios to test the link between genomic offset and establishment probability. And we also want to compare several genomic offset methods. So thank you very much for your attention. And if everything went fine, I should be connected on the Zoom to answer your questions.
Comparing to Beijing, what's the variability if you were to run kind of a similar suitability around the whole native range of the species? Like how much variability is there compared to the variability that we saw in the invasive ranges?
You mean if we take other source environments different than Beijing?
Yeah, for the kind of the full native range. I'm kind of curious what the species spans in its native range compared to where you find it in the invasive range.
Yes, there's a lot of variability because it can be found in whole of China. So there are a lot of different climates and everything. And we tried running the analysis with different source population that were more logical with the invasion story of Drosophila suzukii, because indeed the most likely source population based on the SNPs dataset are not Beijing. So we ran different analysis with different source population, and the variability is quite high, but the main results are still there, meaning that the first invaded areas still have lowest genomic offset, etc., etc.
Cool, thank you. Yeah, I'm very interested in intra region diversity. So that's great. Any other questions?
Hi, I was just wondering, you used all the 19 bioclimatic variables. Didn't you try to remove the ones that are highly correlated? And do you think that the ones that are correlated, because I've seen many papers, they do that. And I just wonder if you did that too. And if not, do you think the correlation between these variables can affect your inferences?
So it's a very good question. Indeed, the method that we use, gradient forest, is supposed to take into account correlated variable with a special algorithm that has been quite studied a lot. There are a few papers about it, and it shows that even if you have correlated predictors, it does not really influence the analysis. So you are able to take all of the predictors. This algorithm allows you to reduce the confounding effect of using correlated predictors.